How's everybody doing today? So we're going to continue on and talk about the electron transport chain. I will hopefully give you about 30 minutes or so for the quiz. So we're not going to have a ton of time. So I'm probably not going to get through all the slides before we take the quiz, but we'll pick up on Friday finishing off the electron transport chain and oxidative phosphorylation and look at my cool ATP synthase video. So we've gone through the process of turning glucose into pyruvate, so the glycolysis, pyruvate into acetyl-CoA, going through the citric acid cycle. And we've made a little bit of ATP along the way, but we've made a set of electron carriers in the form of NADH and FADH2. Those electron carriers, we're going to talk about today how those electrons are then deposited on an electron transport chain, how they go through the different molecules of the electron transport chain, and then hopefully get to a little bit of oxidative phosphorylation when it's the payoff in ATP actually occurs. So first we'll need to talk a little bit about the mitochondria again and remind you guys about the structure of the mitochondria. So here we have our mitochondria, and it has our two membranes in it, and this slide from your book kind of goes through general information about each portion of the mitochondria today, or that goes through both talking about ATP synthase and where things are located and what is in the matrix, et cetera. It gives you a little bit more of the idea of where things are located in the cell. So things like magnesium, calcium, potassium, ATP, ADP are almost free floating within the middle of the mitochondria, things like that. So just to remind you about the structure of the mitochondria, the double membrane leads to what we consider 4 distinct compartments of the mitochondria. So first being the outer membrane. In general, the outer membrane, the outside of the mitochondria, is relatively porous. That allows metabolites to come in and out of the mitochondria. The intermediate space, inner membrane space. So the membrane, the space between the membrane on the outside and the membrane on the inside, which is in essence very similar to the idea of a gram-negative bacteria, where you have the outer membrane of the bacteria and the inner membrane of the bacteria, and there's a space between the two. The environment in there is similar to the cytosol, and it has a very high proton concentration, and we'll talk about that proton concentration and how that gets created. So in essence, a low pH in that inner membrane space. Then the inner membrane, which is relatively impermeable, and that's where the proton gradient occurs that is created from the electron transport chain. The complexes, the complex of proteins that we'll talk about today, the members of the electron transport chain, are located in this inner membrane. Okay? It has these little convolutions in the inner membrane that helps increase the surface area, so the amount of space where these membranes can be located in the inside of the mitochondria. And then the middle of the mitochondria, this is the matrix. This is where the citric acid cycle is occurring, most of it, although we'll talk about one of the proteins, the enzymes in the citric acid cycle, and how it is actually part of the electron transport chain. Then it has a lower proton concentration that causes that gradient than the inner membrane space, so therefore it has a higher pH in the inside. Okay. So we've got the outer membrane, the inner membrane, the space between the two here, and then the matrix in the middle. And my picture is not as good as the, showing this actually what's happening in the cell. You can see some pictures of the mitochondria. You can see these inner membranes on the inside. So we'll specifically be talking about the electron transport chain, which the complexes are located on that inner membrane. Just putting up this picture again to give you kind of a better idea of where things are located. Glycolysis is occurring outside the mitochondria. Coming into the mitochondria now we have the citric acid cycle. And specifically this step right here, which makes the FADH2, is actually part of the electron transport chain. In most pictures you see in biology textbooks, you don't really see them connected together. But one of those enzymes, the one that makes the FADH2, is located within the electron transport chain. And I have a better picture of that from your book. So really that's hooking the two processes together, the citric acid cycle and the electron transport chain. But those complex of proteins are located within that inner membrane as well as the ATP synthase, creating that proton gradient where the pH is really low here in that inner membrane space and the pH is much higher on the outside. Yeah?
The way pyruvate gets into the mitochondria, is that facilitated?
Yes, and we'll talk about that on Friday. Yeah. It is facilitated and way more complex than just this, of course. Same way with NADH. Okay. So this is a picture from another textbook, I believe this is from Pearson's textbook, where you're looking at the phospholipid bilayer and then the complex of proteins that make up the electron transport chain. So we're looking at these proteins here located in that inner membrane. And there'll be an even more complex picture from your book that breaks down each one of these complexes. So we're going to talk about each complex individually and the different components of each complex and what they're doing in facilitating, in essence, carrying the electrons from NADH and FADH2 through the electron transport chain with a couple of carrier molecules, creating this proton gradient that leads to the formation of ATP. And then at the end of this whole process, dumping those electrons onto oxygen to form water. So let's talk about each one of the complexes. So there's 4 complexes of proteins that make up the electron transport chain. Within those complexes contain a series of different molecules that are electron carriers. And they carry electrons and pass electrons differently depending on what electron carrier there are. By transferring the electrons, that proton gradient is created that ultimately leads to the creation of ATP through ATP synthase. So the electron transport chain is those 4 complexes. Those complexes contain series of electron carriers, and as they transfer those electrons, the protein gradient is created. So each of the complexes, each of the 4, is made up of multiple redox centers. And they consist of either a FMN, a flavin mononucleotide, or an FAD, and we've talked about FAD before, that's the one that gets reduced into FADH2. They have a cytochrome A, B, or C, and we'll look at these structures in a second, and also has an iron-sulfur cluster. So depending on the complex, they'll have a combination of these 3 things. Right? So this table from your book breaks up each one of the complexes, and this is the number 1, 2, 3, 4, and then this is the name given to each complex. And we'll look at those in detail, but this will give you a nice comparison of the 4 of them with their molecular weight, so the first one being the largest, the number of subunits, the first one being 43 subunits, and then the groups involved in passing those electrons. Okay? So you can see there are different combinations. Some are heme, FAD, FML. These are the iron-sulfur clusters that we'll talk about. This last one using copper. Okay. So the first element that they all have is either an FMN molecule or an FAD molecule. So if you look at these two molecules, and I apologize for taking structures from different locations so they look a little bit different, but if you can look a little closer, you can see that this portion of the molecule is really the same on both, okay? So this, in essence, this part is that part there. This has just got a different trunk to the tree. So depending on what complex you are, you will have one or the other of these molecules. And these two molecules, we'll start with F and N, are able to carry electrons, okay? So at this location here in the ring, they're able to carry electrons. Similar with FAD, same portion of the molecule carrying the electrons, okay? So reduction, oxidation of the molecule, same, in essence, reaction, just the bottom part of this molecule is different than the other one, okay? So that's where we are getting the electrons at those two locations within the ring. Okay? So this would be the oxidized form of the molecule, again, oxidized form of the molecule, the reduced form of the molecule, the reduced form of the molecule. So the second component that each one of these complexes share is a cytochrome. So these are some different cytochromes. Again, you don't need to memorize these structures. Just realize what they have in common, okay? So they all have this ring structure with the iron in the middle. Just what happens on the outside is a little bit different. Okay? So depending on what complex you are, you will have a different form of this cytochrome. Okay? And the electrons are being carried by the iron in the middle that's able to be reduced. Then the complexes will also contain an iron-sulfur cluster. Similarly, the iron is able to carry the electrons, and depending on what complex it is, it will pass the electrons through different iron-sulfur. And on most of the time when you look at that in your book, it'll just abbreviate them as this. And it won't be specific on which iron-sulfur cluster it is. Coenzyme Q is another molecule that's important in the electron transport chain. And what it does is carry the electrons from one complex to the next. So it's able to, if we're looking at the membrane and we have our four complexes, I'm not doing the picture justice, the Q molecule is able to carry the electrons from one to the other. And as we go through each one of the steps, you can see where it is involved in that carrying. So coenzyme Q is able to pick up the two electrons through this mechanism and carry them from one complex to the next. So in essence, you can think of the electrons as a hot potato. They get delivered onto the electron transport chain through either NADH or FADH2. We'll talk about why those are different in a second. Then within that molecule, they're carried through different molecules, transferred from one molecule to the next, hitch a ride onto coenzyme Q, then delivered to the next, where they are passed along, and passed along. It's a very elaborate chemical game of hot potato. So the coenzyme Q is able to readily accept those electrons. It also in the process picks up two protons, and it can freely diffuse in the membrane. That's the importance of coenzyme Q. It can diffuse through the membrane in between the two complexes. So in the relay race, the coenzyme Q is actually the runner. So in this picture, again, here's our complex 1. The electrons are then being distributed to Q running through the membrane, distributed, and then we'll talk about another carrier, cytochrome C. Okay? Again, NADH and FADH2 will deposit their electrons on different complexes, and we'll talk about the reason for that in a second. So this is a nice, good, generalized picture of the electron flow. We're going to go into each one of those specifically in detail. Now, you're going to notice by the fourth complex that everything is very complicated. I am not expecting you to memorize precisely where each of the electrons are being distributed as they move through each complex. But just realize that they are being transferred through these different molecules, either an FMN, an FAD, through the cytochromes, through the iron sulfur clusters, and then the coenzyme Q is able to move them through the membrane, okay? So you'll see an overwhelming amount of information in each one of these slides. I'll try to help you guys focus on what you need to memorize, for lack of a better term, in this process. So let's first talk about complex 1. This is the largest one, and it's one of the largest assemblies of proteins and molecules in mammalian cells. There are over 43 in that table, different polypeptide chains, and these are proteins that are encoded either by the chromosome or by mitochondrial genes, the DNA located in the mitochondria. The binding for NADH, so the binding site for NADH is located on the matrix site, so the internal portion. There are FMN molecules that are non-covalently bound that accepts the 2 electrons from NADH. So, in complex 1, I don't know how the eraser ends up back there. Just like if I needed to time out, just like I need a second. Just gonna go behind the screen. Okay, so we have our first complex. Again, my picture is not as good as the book. So on the matrix side, the NADH is going to first transfer its electrons to a molecule of FMN that is non-covalently associated with complex 1. Then within the complex, some iron sulfur centers pass the electron throughout the complex. I will show you your book's picture. That's much better than my drawing. Actually, this is another book. This is from the biochemistry book. It's actually more simplistic than the one in your book. So here's the NADH passing its electrons to FMN. Then the FMN is transferring the electrons through these iron sulfur clusters. Okay? Then coenzyme Q picks up the electrons and is able to transfer those electrons through the membrane. Okay? So just going back to the molecules we talked about, so this is the FMN, picks up the electrons. They're passed through the iron sulfur clusters, so from one to the next, to the next, to the next. Then they're picked up on coenzyme Q, which is able to carry the 2 electrons along with 2 hydrogens, and then can disperse them through the membrane. On the other picture I showed you, it was on this side. Semantics, just different drawings, okay? So, picks up the electron from NADH. NADH is from the citric acid cycle, right? Drops those electrons on FMN, goes through the iron sulfur clusters, coenzyme Q picks it up and is able to diffuse it through the membrane. During this process, hydrogens are also pumped from the matrix into this inner membrane space. Okay? So far, the experiments that we have been able to conduct show that should be about 4 hydrogens being passed from the matrix to the intermembrane space. That number is actually relevant because how many hydrogens are passed will determine in the end how many ATPs are actually made. So there's a little debate on how many hydrogens are actually transferred from the matrix to the intermembrane space. As you can imagine, studying a organelle and what is occurring in the organelle is quite difficult. And looking at the movement of hydrogens. So a lot of this work is done on bacteria, and we're making inferences about what happens in the mammalian cells or in the eukaryotic cells. Okay, so that was complex 1. Complex 2 is succinate dehydrogenase. I have exactly 3 minutes to go through 2, and then we're going to stop. So complex 2 accepts 2 electrons from FAD. Well, okay, let's go back into the citric acid cycle. So complex 2 actually contains this enzyme here that's important in the citric acid cycle, succinate dehydrogenase. So succinate dehydrogenase, that's responsible for changing succinate to fumarate, is embedded in the mitochondrial inner membrane. That is the only one of these proteins, these enzymes, that's embedded in the membrane. So that links the citric acid cycle to the electron transport chain. And this formation of FADH2 goes directly into the electron transport chain. So succinate to fumarate from the from the citric acid cycle makes an FADH2. So because this enzyme is located within the electron transport chain, that FADH2 molecule immediately transfers its electrons through the complex to, again, coenzyme Q. So unlike NADH that goes on the first one, in complex 2, the enzyme that actually changes succinate to fumarate and makes FADH2 is located right in the electron transport chain. So again, we're picking up hydrogens on FAD to make FADH2. We are carrying those electrons through the complex 2 through a very complicated pathway. Okay, we'll stop there. You guys go ahead and put your stuff away. Does anybody have any questions before we start the quiz? How you do on this quiz should give you a strong indication of how much additional study you will have to be doing for the exam.